Hi, everyone. Welcome to our research project presentation for the Think Neuro 2023 Summer Research Program. It was an honor to be selected for this program, and I'm very excited to share our work and findings with you. In this presentation, my team and I will be going over the results of our bibliometric and network analyses of the top 100 articles on Rett syndrome. And before we jump into the presentation, we wanted to introduce ourselves to you. My name is Parinita Balaji. I'm the Research and Conference Associate for the Neurosurgery and Neurodevelopmental Disorders team. I'm also one of the co-facilitators of this project. I'm currently a rising senior at Amador Valley High School, which is in the Bay Area.
Hi, I'm Sydney Munyon. I'm the other co-facilitator of this project, and I'm currently an undergraduate student at Tulane University.
My name is Stephanie Hung, and I am a rising junior at Monte Vista High School in the Bay Area.
I'm Alicia Joseph, and I'm a rising junior at Amador Valley High School.
My name is Rayleigh Cheong, and I'm a rising sophomore at Northwood High School in Irvine.
My name is Jolene Wong, and I'm a rising senior at Marquepo High School in Alhambra.
Hi, I'm Jolena, and I'm an incoming junior at Franklin High in Belmont.
I'm Sarah, and I'm a rising junior at UC Berkeley.
Cerebroatrophic hyperammonemia, otherwise known as Rett syndrome or RTT, is an intricate and extremely rare neurodevelopmental disorder caused by mutation on the methyl CpG binding protein 2, otherwise known as the MECP2 gene, found on the X chromosome. Rett syndrome primarily affects females, however, it can also affect males. Beginning from diagnosis, which is typically done in the early months or years of a child's life, this disease impairs nearly every aspect of life. The MECP2 protein is vital to the regulation of gene activity and depth of synaptic connections between neurons, and with a mutation in the gene that provides the instructions for this protein, many motor and sensory processes are inhibited.
So in this first figure, we're kind of just showing how we selected and then extracted all of the data that we used for this project. So we used the online database Web of Science to find academic articles about Rett syndrome. We used one search term, just Rett syndrome, which produced just a little over 6,000 results, which is obviously way too many articles for us to use and go through in just this one project. So we sorted all of these by number of citations, with the highest being first, and took the top 200 articles with the most citations for Rett syndrome. Then of these 200 articles, we further looked through them and used the inclusion and exclusion criteria you can see on the slide to select which ones would be appropriate for our uses. And it came out with a total of 114 articles. So we decided from there to narrow it down to just the top 100, still being sorted by most cited, and took these straight from Web of Science. They were extracted as both an Excel file and a plain text file so that we could use them and map and analyze all of the data that you'll see later in this presentation.
This first table shows the top 100 most cited articles on Rett syndrome sorted by highest to least. The top 100 most cited articles on Rett syndrome were published between the years of 1985 and 2016, of which 33% were published after 2010. In total, the top 100 publications were cited 46,968 times, ranging from 191 to 4,371 citations, with the average number of citations being 469.7. The most cited article at 4,371 citations was Epigenetic Regulation of Gene Expression, How the Genome Integrates Intrinsic and Environmental Signals by Jaenisch et al. By observing the data, it can be derived that the most productive year was 2005, with 11 articles in the top 100 list and an average of 305.7 citations total.
From looking at the data extraction of the top 100 articles on Rett syndrome, there were a total of 71 first authors who published these articles. And of those 71 authors, this figure focuses on the 13 first authors who published 2 or more articles. The top 3 authors on this figure were able to contribute to a high number of total citations, with all of them exceeding 2,000 citations. And aside from these top 3 authors, there were a total of 3 first authors of the 13 who published 3 articles, while others only published 2. These authors include Guy Zhang with a total citation of 2,303, Moretti P with 873 citations, and Hagberg E with 805 citations.
So here we have a bar chart representing the distribution of the publishing institutions. And of the 48 different institutions that contributed to the top 100 articles that we considered in this study, 12 of those institutions published 2 or more articles. And the top 3 institutions were the Baylor College of Medicine with 19 articles and 11,593 total citations, the Massachusetts Institute of Technology with 10 articles and 7,787 citations, and finally, the University of Edinburgh with 10 articles and 4,211 total citations. Among the 12 institutions which contributed 2 or more articles, the average number of citations was greater than 200, with the University of Texas Southwestern Medical Center having the highest mean number of citations at 949 times.
This table lists the journals in which the articles were published. The top 100 articles on Rett syndrome appeared in 40 different journals, where 40% of the articles were published in just 5 journals. Neuron and Human Molecular Genetics were the top 2 contributing journals, with 4,319 and 2,747 citations respectively. Nature Genetics had the highest citation count, with a total of 10,919 citations. 24 of the journals only published 1 article on Rett syndrome, but notably garnered 8,298 total citations and a mean of 345.8 citations for each paper.
Figure 5 shows which countries the top 100 most cited articles were published in. These top most cited articles on Rett syndrome were published in a total of 13 different countries. The USA, UK, and France were the top 3 contributors, publishing 61, 15, and 6 articles respectively. Belgium, Italy, and Canada contributed equally, each publishing 3 articles. Australia and Sweden both published 2 articles, and China, Switzerland, Spain, Germany, and Israel published 1 article each.
This next figure displays the distribution of research themes of the 100 academic articles in a pie chart. In total, the 100 articles on Rett syndrome and autism spectrum disorders covered 17 different research themes. The top 4 research themes were neurosciences and neurology at 29.9% of all articles, genetics and heredity at 19.5%, and biochemistry and molecular biology and science and technology other topics tied at 10.4% each. The total articles were 23, 15, 8, and 8 respectively. As Rett syndrome is a genetic mutation affecting the brain, it is no surprise that neurosciences and neurology and genetics and heredity were the 2 most common research themes.
This chart shows the word's frequency over time in the articles. Occurrences of many of the keywords have significantly stagnated in recent years, such as the words CpG binding protein, gene, expression, and mutations. Overall, it seems the only keyword to gain frequency is Rett syndrome. Along with Rett syndrome, the words CpG binding protein and mouse model have the top 3 cumulative occurrences in the articles. The keywords that have stagnated the least refer to more specific and unknown aspects of Rett syndrome.
For example, mouse model, which refers to a laboratory mouse used to study human disease, and DNA methylation, a major cause of Rett syndrome that was only recently discovered, are some of these keywords. These keywords signal a shift in the focus of research of Rett syndrome, indicating that researchers are now looking at the causes of Rett syndrome and are attempting to find cures.
This biblioshiny figure shows the co-occurrences between topics through a network map with different colors. The most common links were found between Rett syndrome in blue and CpG binding protein 2 in green from the 100 articles listed in figure 1. Within Rett syndrome, mouse model and DNA methylation are the largest common ideas, as mouse models are used to test for Rett syndrome treatments, while DNA methylation is an epigenetic mark pivotal for understanding the genetics behind Rett syndrome. There are also smaller co-occurrences with histone deacetylase, enzymes that remove acetyl groups, as seen in purple.
This graph shows the average trends of the annual scientific production rates on Rett syndrome articles. As the years progress, we can notice a peak in the scientific production roughly every 5 years. By looking at the chart on the left, in 2002, there's an increasing trend in productions until 2005. Then the rate starts to decay and the pattern repeats. Looking at this figure, the article production rates on Retts noticeably correspond with their patients, as symptoms tend to appear later on in their lifetime. Patients with Retts are often diagnosed from ages 6 to 18 months, and they gradually show stronger symptoms as they age. The consistent upward trends in article productions are a complement the time expectancy of symptoms to show in these patients.
So in this bibliometric study, we studied the top 100 articles on Rett syndrome or cerebroatrophic hyperammonemia using many tables and charts that we designed, as well as those that were constructed using bibliometrics and biblioshining software. Using the results of this study, our team concluded that research on Rett syndrome has seen significant growth in the past few decades. Researchers predominantly in the United States are now focusing on the genetic basis of disease, such as the CpG binding protein that was referenced in this presentation, in order to better study Rett syndrome's progression in patients, as well as to develop potential therapeutics to combat this disorder's symptoms. And according to our literature review, many research papers are now publishing the results of mouse models and of clinical trials for potential Rett syndrome drugs. To conclude, this study was of great significance. It shows how research in Rett syndrome will only continue to be of interest in the upcoming years, as the scientific community continues to work towards developing a cure that can change the lives of young girls and young boys struggling with Rett syndrome. We hope you found our research to be as exciting and informative as we found it to be. Thank you so much for your time and interest in our project.